From Microbe TV, this is Immune, episode number 27, recorded on January 8th, 2020. Happy New Year, everybody. I'm Vincent Racaniello, and you are listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hello again. Happy New Year.
You too. And from Durham, North Carolina, Steph Langel.
Hey there. Yeah, good to be back. 2020. Got over some sicknesses, feeling healthy.
This is our first cast of the new year and our first cast of the new decade.
Yeah, how about that?
This could be our first full decade of Immune, right?
Wow.
Well, that's exciting.
Who knows?
Wow.
I'm up for it.
Steph will become a professor and say, I don't need you guys anymore.
No,
Don't leave us.
definitely not. The biggest thing is that if I just get my own office in the next decade, then we'll be set.
That would be great.
That would be
Even though you don't have one, it sounds
Right.
good. Although I
good.
understand for you, it's a hassle, right? You have to
Yeah, it's
run
just
across
trekking.
campus. Yeah.
Yeah. But I mean, it's not, I shouldn't complain. We do have a sound studio and it's nice and it works. So
Okay. Well, if you like what we do here on Immune and also our other science podcasts, consider supporting us. Microbe.tv/contribute. We'll talk a little bit more about that later. But right now we have a paper to start your new year off. And Steph is going to give it to us.
wow, a lot of pressure. New paper,
Cool
new
paper.
year.
It's a very cool paper. I have to say,
It really is.
of all the immunology papers that we have tackled, I understood most of this one. I don't know why. Maybe because it's 2 years of Immune now, right?
Well, it could be a little osmosis through Immune. But I do have to say, one of the reasons I chose this was I felt it was very digestible. It's not easy science. These are difficult concepts, but I felt that they wrote it in a way that made it.
And here's the thing which I really like. At the end of every experimental section, they write the conclusion.
Yes.
And not everybody does that. Not everybody does that, right? And I think it really helps because then you could go back and say, oh, that's what that experiment, so it helps.
Yeah. And there are diagrams for the different schematics, which is great.
And great.
That was these were some complicated experiments that we're going to go over.
Right. Yeah, and I actually found out about this paper via Twitter, as I find out a lot of new papers that pop up there before they do on my email for my PubMed subscription. So I like that aspect of Twitter. And because of that, I follow the main author, and now I follow the PI. So I was able to interview the lead author and get a little insight into his background. Yeah. And I asked some specific questions about the paper that helped clarify. So I'll kind of pepper his responses in as I go through and explain what they did and why they did it. So that should be a little fun aspect of this as well. So I'm starting off the new year really taking the dogma of immunology and what we consider innate versus adaptive and kind of busting it for our audience. Yes. As immunologists, this has been something that we've seen probably over the past decade, peppered into the literature here and there. But I think it speaks to the grander trend in science that we have new editions of textbooks for this reason, to update with updated literature. So the immune system, as we know it, the adaptive part of it, the innate, is the quicker responding part. It's supposed to respond to pathogens quickly and kill infected cells or otherwise abnormal cells like tumor cells. And then the adaptive part is an ability to remember a previous encounter with an antigen. And the innate cells don't, asterisk, have that response. And it's the stronger response upon the secondary encounter with faster kinetics that really characterizes this response. And so we know that as immunological memory, and it's the basis for vaccination, and we're trying to harness that for cancer and many other different types of things. But we've known it specifically as a B and T cell response. This is specific to B and T cells. These adaptive lymphocytes, they're highly specific. They have somatic diversification mechanisms because of genes that can rearrange the genes of the receptors of the B and T cells. And those responses of the receptors and the antigen really set up the initial response. And then, of course, the more robust response down the line. But the problem is, what happens when a paper that describes an innate immune cell having this type of increased response to an antigen the second time? We kind of have to take a look and try to understand what this means. So the very first evidence that NK cells, and I'll describe NK cells in a bit, but the very first evidence that this innate immune cell, natural killer cells, has somewhat of a memory was in 2006. So we're past a decade ago. But what they demonstrated was there's a specific subset of natural killer cells in the liver that were able to mediate contact hypersensitivity to two different chemicals. Contact hypersensitivity we thought was mediated alone by B and T cells, but they showed that mice deficient in B and T cells had this hapten-specific, haptens are small molecules, contact hypersensitivity response. And they went on to do adoptive transfer studies to find it was the NK cells, the innate natural killer cells that are supposed to be these quick responding, A-specific, non-specific cells that demonstrated this recall response. And
for those people who aren't familiar, this contact hypersensitivity, do you have examples of what causes that so people can put that in their mind?
Yeah, sure. I mean, they do it a lot with allergens when they're trying to test your recall response. You know, if you go to the doctor and you are trying to determine if you're allergic to something, that I think TB is a contact hypersensitivity test.
Yep.
It's really small molecules that you're trying to see if you have a recall response to, and it's supposed to be mediated, I think, mostly by T cells. But
yeah, so I think poison ivy is also considered contact, hypersensitivity, right. And nickel allergies or things like that. So a lot of skin related things.
Yeah, and it's kind of a quick way to look at a memory, quote unquote, response that doesn't have to be based on antibodies, where you have to do serology. And so that was kind of the first evidence that, wow, maybe these innate cells are doing something with some recall or trained memory, as I'll describe throughout the paper. So a little bit about natural killer cells. I mean, as I described them as innate lymphocytes, that's because they are, when we have a stem cell, they divide into two different lineages. There's myeloid lineages and lymphoid lineages. And myeloid cells are a lot of the innate cells, the quick responders. And even though NK cells are traditionally these founder innate immune cells, they are actually on the lymphoid, on that lineage. So they're in the same family as B and T cells. So that's an interesting thing to keep in mind when we're talking about similarities with NK cells and maybe this memory response. Mm-hmm. But natural killer cells, I mean, they respond to cells that are infected with viruses. They respond to tumor cells, and they respond rapidly by secreting perforin or granzyme. They can kill infected cells, but they also secrete a lot of cytokines like interferon gamma, which can recruit other cells to the area and also further enhance killing of target cells. And they do this by recognizing stress-induced ligands on virus or tumor or virus-infected cells. But it's kind of a delicate balance. Their receptors, they have both inhibitory and activating receptors. It would take a while to go through all of those, so I'm not going to. But I'm just going to say that their activating receptors are the ones that can recognize the stress-induced cells, where their inhibitory ones are going to engage with MHC class 1 to try to recognize our own cells, so we're not killing our own cells. So thinking about natural killer cells as maybe having this memory, or innate cells in general, there's been other evidence of innate cells having this recall response. More recently, I think macrophages have been demonstrated to have some type of recall response. So, and really when we think about the evolution of immunology, it's not that crazy that innate cells might have this type of memory, because when you look at some of our earliest primitive vertebrates, and those would be the jawless fish, which I think we did an episode on lamprey a while back.
Mm-hmm. Yeah, we did.
People can look at that. I mean, they evolved before lymphoid cells with diversified receptors, and they've been demonstrated that they also can mount this recall response. So it's not that crazy of a suggestion, and it's probably why people, started looking at this in the first place, in trying to understand, well, NK cells and innate cells, maybe they do have some type of recall response.
So, Steph, you know what my connection is with NK cells? A very tenuous one, but
What is is it to do with polio?
So the poliovirus receptor is an NK cell recognition molecule.
Oh, interesting.
Which was found out by other people many years after.
Sure.
We had done it. So it's funny.
Yeah, no, that is interesting. And well, that leads into thinking, so over the years, I guess over the past 10 or so years, when they're trying to define the different subsets of so we're calling them memory-like NK cells, they've identified 3 different kinds, which relates, Vincent, to what you're saying about viruses. So one of the kind, they call it, so cytomegalovirus, mouse, specifically cytomegalovirus, there's a glycoprotein on its surface that is recognized by a germline encoded receptor on NK cells. And so that's kind of like the viral encoded ligand interacting subtype.
You know, what's funny is that so apparently the CD155, the polio receptor, is involved in NK cell recognition of CMV infected cells because CMV infection downregulates CD155.
Right.
So that it's not, the infected cells are no longer seen by NK cells.
Right.
Isn't that
Look
cool?
At what they're up to. It's very cool. It's very cool. What
viruses are very sneaky in modulating the immune system.
You have to be sneaky to work on them too.
Yeah. So there's like this viral adapted memory, and then there's what they describe as just antigen-specific responses. So I just kind of described like haptens or small molecule-specific responses. But then there's cytokine-induced antigen-independent memory-like responses where activating cytokines can kind of promote the differentiation of NK cells to respond more quickly later on. So it's out there and it's interesting, and it's not as well defined in terms of why they matter, but why people care is because if we can train NK cells, they're really important potentially in cancer. So NK cells can kill tumors, obviously virus infected cells. And so if we can harness this activity, potentially could be used for treatments. And so I think the cancer world is where a lot of the focus is right now. But the paper we're going to talk about, they don't focus on a cancer model. They actually focus on a totally different model that, from what I understand, really is under investigated in this NK cell memory field. So before I move on, do you guys have any questions about NK cells? And
are there diseases of people with defects in NK cells? There must be, right?
There must be.
I can't think of anything.
No.
Not off the top of my head, but if I do a quick search, they're saying
there are mutations in GATA2, which is like a core transcription factor, and that causes human NK deficiency.
Okay. Yeah. So there's
but I'm not sure what the outcome of that disease would be. You know, it's interesting with deficiencies because certain immunodeficiencies cause really profound disease outcomes, but then others are not quite so profound because there's, redundancy in the immune system.
Yeah.
And I'll tell you that for a long time, there wasn't much studied about NK cells.
So the article here, NK cell deficiency, maybe you landed on it as well. It's a Journal of Allergy and Clinical Immunology. It's by Jordan Orange.
Yeah, I'm reading that too.
He just moved here.
Oh, wow.
Oh, really?
Yeah, he's here. He's working. That's right. I remember hearing him talk.
He's huge in the NK field.
Yeah.
You can call him up. Yeah. It seems like obviously susceptible to viral infection, most notably papillomaviruses, herpes viruses. So with this paper, let me get back to my, yes. Okay. So, I'll give you the title and then we're going to go through the paper and you'll be able to understand kind of what the breakdown of the words are. But, so I'm going to give the long name. So it's histone H3 lysine 4 monomethylation supports memory-like NK cells induced by systemic inflammation. And so the abbreviation of that long beginning histone 3, H3 lysine for monomethylation is just H3K, which is the letter for lysine, K4ME1. And this is published in Cell Reports, so it is open access. You can check it out for free. You could follow along with us if you wanted to. But this is coming out of the Melanie Hammond lab. She's at the Institute Pasteur, and their group looks at chromatin biology, epigenetics, and innate immunity, obviously the NK cells. And the person who is the first author, his name is Orhan Rasid. And I was able to get a little background on him and what he was doing before he came to Melanie's lab. And so I'll just give a little kind of brief synopsis of what he was up to. So he trained as a veterinarian in Bucharest, Romania, and then kind of started working in an immunology lab while he was there. After he graduated, he did a short-term fellowship at Utrecht in the Netherlands, and then he was on to Pasteur Paris University for a PhD program. And he started in the lab of Jean-Marc Cavaillon, which is
Cavaillon, I think.
Cavaillon, thank you.
Yeah.
Which is also on this paper.
Yeah, also an author. Mm-hmm.
Yep. At the Institut Pasteur. And then after that, he joined Melanie's group to work on epigenetics like memory NK cells. And so really it was his PhD that got him interested in NK cells. And then looking at memory with NK cells was kind of just a natural step for them because I think they're interested in epigenetic changes, which we know from previous literature play a large role in this trained immunity of innate cells, that modifications like methylations or and histone acetylations, that's kind of driving this. So that was the impetus, I think, behind his work here. So starting with the paper, they wanted to set up a model of systemic inflammation. And so what they did was they used mice in an endotoxemia model where they're giving the mice either PBS or they're giving lipopolysaccharide, which is a part of E. coli. So you can just give pure LPS and it's going to induce acute inflammation, but low mortality. So they showed the mice after they're giving LPS, they don't die. They have full clinical recovery by day 7, but you can see they do lose weight. They do show signs of inflammation. They're usually, these clinical scores, the mice are very sad looking. They're disheveled. They're usually hunched in the corners. And so this is the model that they wanted to use. There are other models of systemic inflammation and sepsis in particular. I'm thinking of cecal ligation puncture, which is more of a polymicrobial, I think. They wanted...
Yes.
Right. Yeah. So cecal ligation puncture is, I think, a better model of sepsis in terms of cytokine release, but it's not very specific. And I think what this crew wanted to do was look at LPS specifically and what it's doing to the NK cells. So that was their model. And previously they had, I think, worked with this model to define NK cells and what they're up to in the acute phase. And acute, when I say that, would be like 48 hours, 24 to 48 hours. But thinking of NK memory-like responses, they wanted to look at these after 14 days. So they took mice, they injected with PBS or LPS, and then they followed them for 14 days. And they took out the NK cells and they profiled them. When we profile cells, we can look at the surface receptors to define phenotypes. And then we can look at function by what they express. And so what they did was they phenotyped, and they didn't really see differences in splenic NK cells after 14 days post either LPS or a PBS injection. But when they went on to look at function, they looked at interferon gamma secretion, which is a main function of NK cells. And they were able to find that those cells that were given from animals given LPS at day 14 had a greater percentage of interferon gamma producing cells. Let me go back actually. That was when they took them out. So I want to say that when they're inside the context of the spleen, so before they purified the NK cells, they looked at it within the environment of the spleen, and they actually showed that the response was lower. So that's opposite of what I said. And the reason why that's important is because the spleen after LPS, after inflammation, is going to tamp down the immune system. It's going to increase T regulatory cells. It's going to increase certain types of myeloid cells. And so what they saw was a lower interferon gamma response within the context of all the cells in the spleen. However, when they stimulate the cells with cytokines, that they were responsive. So this wasn't a factor of the NK cells are not responsive. It was a factor that they were in an environment that was not conducive for them to respond, probably because of Tregs, which they saw went up after 14 days. And so what they did do after that was they took those cells out of their splenic environments and they just said, okay, let's look at NK cell intrinsic interferon gamma secretion. And it was there when they pulled them out away from that immunosuppressive environment that the LPS stimulated had higher amounts when they were further stimulated with LPS.
So this, because it's 14 days, they consider this a memory property, right?
They are considering, yes. So they are considering
is that long enough? I mean, it doesn't sound like memory in terms of the classical, T and B cell memory.
30 days would be memory, right?
Yeah. Yeah. And I think I'm glad you brought that up, something I wanted to discuss when we're defining memory. I think with these innate cells, we kind of have to alter the way we think about that. This is definitely past the acute phase. Yes. But and we'll learn in a little bit that they do look beyond that. They look at, I think, 9 weeks.
Okay.
But yeah. But what they're looking at here is that when
9 weeks, right? It still persists at 9 weeks. So.
Still persists at 9 weeks. But but when they're looking at 14 days, they pull them out of the splenic environment, stimulate them with activating cytokines and LPS, and they do see a response only in the mice that were previously given LPS. And so if you have a response only in those mice, maybe they could start to suggest that these are memory-like NK cells, because otherwise the response would be similar between LPS and PBS injected mice.
Mm-hmm. I guess.
Go ahead.
I guess this would stimulate some human studies now, where you would take people, say, who have influenza or CMV, whatever, and then at monthly intervals take out NK cells and see if they can respond again to viral antigens, right?
Yeah, definitely. I mean, and that would be interesting to see, because these experiments are they're looking at a schedule that's very defined.
Yeah.
And those viral infections, it's hard to say when, depending on if the person was recently infected or not. So human studies, I think, would definitely be the next step.
There are a couple of viruses that we can use to infect people. So you can use vaccine strains of flu, you can use rhinoviruses, and et cetera. So there are a few where you can do challenges, and that would be, that way you could have a defined period, right?
Right. So I was going to say that the only downside to this whole paper that I had is when I looked at this figure 1 and I looked at this, the whole premise of the paper is that these NK cells are responding more robustly with interferon gamma production if they are isolated from mice that were pre-challenged with LPS versus PBS.
Correct.
And the differences are really small.
Yeah.
I mean, they are really small.
Which, yes, we've talked about in previous episodes about, when you're looking at flow cytometry differences, what is biologically relevant? Okay, yeah, there are 3 stars here when I'm looking at, which indicates highly significant, but and they do bring that up. They say that most of our differences are within 10%, but I think we'll learn at the end of the paper when they challenge these mice with bacteria that it did matter.
It did matter. I agree.
Biologically. But you're right. You're right.
These are, I mean, I sit and look at this and say, if my student brought me data and that was the graph, would I look at that and say, oh, we have a whole story based off of this? And I'm not so sure.
Right.
Because I've looked at a lot of flow cytometry plots, and that would not strike me as something that was something to chase after.
Right. And also because these mice, these are inbred, highly defined mice. And so to show this in a population like that's outbred, like humans, or you could do the wild mice, the outbred mice that we've talked about in the past, and see if this still persists. But no, it's a great point. And it's something that I think we battle with maybe in these mechanistic immunological studies. Yeah. And then how do they translate something I think we can.
So on a recent TWIV with Chris Sullivan, he was saying, virology is used to 1 log minimum differences, right?
Right.
But in the world of microRNAs, you get 2 or 3-fold effects of microRNAs, and that's what we have to live with, right?
Yeah, right.
So that may be the same thing here.
Yeah, right.
Yeah,
like, this might be what to live with.
Don't get me wrong. There are small differences that we would analyze, and they would be really interesting, but when you're so for example, I've seen some immunology papers where they're talking the difference between like 0.01% and 0.02%
like those rare
of cells in a population. And I'm like, I can't even gate cells to consistently give you a 0.1% difference,
Yeah,
and so I really start to struggle with some data that are out there. And, this one, I guess if you do it lots of times and it repeats and you're really convinced, and then there really is a difference. But even the gamma production, it's modest difference.
Yeah. And that was a question I had asked. I don't think because this is a follow-up, I had asked him, what about maybe other output assays? So,
right.
Perforin they're not looking at cell lysis. That's not their model. So I understand why they didn't do, they just stuck with interferon gamma because it's related to sepsis. But it would be nice to kind of have more robust output. But I also get sometimes you just got to get it out there. You got to get.
Oh no, I agree. And and I'm not trying to knock this paper, but I'm also
For sure.
if we have trainees out there too, if you have a small difference, maybe it's important, maybe it's not. But maybe you need to repeat it more than twice and see if it's a really consistent but a very small difference. It could still be important.
Right. So I think with this kind of first figure, this first section is really defining their model and then looking at these cells both in vitro and then pulling them out and looking at them ex vivo. We can summarize it kind of three different bullet points. So the surface phenotypes were not different between LPS and saline treating cells at day 14, but they're showing maybe modest functional differences in interferon gamma, which the trend switches, and my apologies for jumping to the last part when I started explaining this, but the trend switches depending on where the NK cells are. So if they're in the environment of the spleen, at least in this context, they had decreased interferon gamma secretion after they were stimulated with LPS. But if you can pull them out of the splenic environment, then it flips. And so I think what it suggests is that these cells are responsive, and this field, which is another thing Orhan said, was that how do you release the suppressiveness of the splenic environment? Because we can't. They're always in context. All of this is in context with the cells around it. So that led them to say, okay, let's look in vivo. And I think they set up their model based on this difference between endogenous within the splenic environment and then intrinsic, what the NK cells alone are doing. I will bring up they did kind of tell us that in these original experiments, the cell purity of the NK cells was only 80%, which is low. And so you could have some contaminating cells that give you different results. But they use the mouse model that we're going to talk about for the in vivo studies. Then they're called congenic mice, and they're used a lot in immunology. And they're CD45.1 and CD45.2 mice. The only difference is that one strain expresses the 0.1 allele while the other expresses the 0.2. And so they're the same asterisk, because there's actually something about these mice that are different, which I'll bring up later. But you can detect differences in the cells based on, and you can by antibodies that bind to either 0.1 or 0.2. So they use those mice to try to further clarify that, yes, the first experiments were done with 80% purity, but when they co-culture with a highly pure population, the results are the same. So it's kind of a redundant way to tell us that what they're seeing is true. So they went on to the in vivo experiments. And what they first did was explore the memory of these NK cells after LPS challenge. And so in order to compare these day 14 LPS NK cells with naive counterparts in the same immune environment, they used an adaptive transfer model where naive congenic NK cells were transferred into post-endotoxemic mice. So what this means is they took the CD45.2 mouse, they either challenged it with PBS or LPS, so just like I described before, and then they waited until day 13, so one day before they're interested in re-challenging them, and then they gave them naive cells from the CD45.1 mouse. So those NK cells can be distinguished because of the 0.1 and 0.2, but now you have endogenous cells that have seen LPS or PBS as control, and in the same mouse, you have adoptively transferred naive cells. And so then by looking at transferred naive endogenous PBS treated and endogenous LPS, they can ask the question, is in vivo, do they see the same kind of trends? And so when they do that, they gate and they later then look at interferon gamma secretion again. So it's kind of like their standard output assay. And in the animals that were originally treated with PBS, both the transferred and the endogenous cells, they have the same amount. So that shows that adoptive transfer didn't make a difference in what the interferon gamma secretion was doing, which is good. We want that to be consistent. But then the only difference was the transferred naive NK cells were significantly lower in their secretion in the mice that were originally given LPS compared to the endogenous LPS treated cells. So basically what that meant was that the endogenous LPS cells had a memory response to the LPS challenge. And so it was a way to, in vivo, show what they had demonstrated in vitro within the environment of the spleen themselves. Because remember, the original cells that were treated with LPS before they were challenged, those were in. So when I say endogenous, it just means in the mouse itself. And then the ones that were transferred were naive. So I think that really showed that, okay, in the surrounding environment. Well, this was interesting because I assumed that, so what they actually showed in the in vitro work was that the splenic environment would dampen the ability of the endogenous cells to respond. But they showed here in vivo that it was fine. They responded. Yeah. So I thought that was interesting.
Yeah, I noticed that too. So it's a little different from what other people are saying, right?
Well, it's different from what they showed, because I think they were saying like, oh, the reason we don't see it is because T regulatory cells, but then when you do it in an animal, it's fine.
Yeah.
Well, I mean, that still might be suppressed, right? Because if you took those same cells out, they still might behave more robustly in their interferon gamma production. But I think that the important comparison is that those mice, those NK cells that were around when there was that LPS conditioning, respond more than the ones that were not around, but they're in the same mouse at the same time.
Right. So that's looking at surrounding environment. And so then they asked, well, let's look at the intrinsic response. So what they did was they took one of the congenic mice strains, they used 0.1. Again, they treated it with PBS or LPS. And then day 13, they transferred in NK cells into the other congenic mice, so the 0.2s. And then they challenged those mice with either PBS or LPS. And so now what they have is an endogenous naive transferred day 14 PBS or transferred day 14 LPS. So before you were transferring in only naive cells to look at the differences. And now what you're doing is you're transferring in cells that have seen LPS. And then you're looking at the differences. And similarly, they were able to show that only the mice that had previously seen LPS, both endogenously and transferred, those were the ones, the transferred were the ones that had the increase in interferon gamma compared to the naive NK cells. So I like those models, those adoptive transfer models. I think it's, redundant's the word, way to say that no matter where the NK cell sees the LPS, they're going to respond with an enhanced response compared to the naive cells. Because I think trying to convince us that this is a memory response is what they're doing here. Would you agree?
Mm-hmm. Yeah.
Right.
Yeah.
That's good. Yeah.
Right. And here is where they describe how they're going to use these adaptive transfer models, and they did the same thing, but they did it at 9 weeks post endotoxemia. So they took LPS and PBS, they waited 9 weeks, and they showed similar-ish. I think that the differences that Cindy's talking about get even smaller. So, when you're looking at and I think they recognize this. So they did a little in vitro section where they pulled out the cells and stimulated them again with activating cytokines plus LPS or just activating cytokines. And they saw an increase only when you give those cells LPS and only from the animals that had seen LPS. It was not increase in the PBS animals. So, it's still significant, but these become even lower. So here's a difference where it's not immunological memory, because we would, in a B or T cell, it would have been greater than the previous, but we're not seeing that. We're seeing it's you may still see some trained memory, but this is not a greater response kinetically.
Let's just say it this way: if you looked for a memory response from a B cell, let's say you immunize a mouse, and then you see the peak antibody response, and then it goes back down, and then you re-challenge the mouse, there's going to be orders of magnitude more antibody production.
Yes.
It's like, there is no question. This is like, there's a few more interferon gamma producing cells. Right. So it's there. But, the flip side, one could argue, is that you wouldn't want that level of memory that you see in a B or a T cell in an innate immune cell, right? Because this is not specific. Because this is going to happen theoretically no matter what infectious challenge you have.
Well, it's interesting. Well, you go ahead.
There's some specificity because this is LPS mediated, so there's some LPS specificity that you'll get to. But the idea that it's generalizable, that like, let's say if this is LPS, it could be any gram-negative. You get a gram-negative infection, you get another gram-negative infection, you get another gram-negative infection. This could lead to overwhelming systemic problems, excessive inflammation, right? And cytokine storm and death, potentially. So, this is a really tricky system to develop memory to. Like, you want a little bit better, but how much better?
Right. Now,
are we only able to see this because this is a mouse model where these mice are housed in like extraordinarily clean conditions, and then we challenge them with something and you see something.
Well, you're right. Because, we are seeing LPS at all hours of the day, all the time. It's just a matter of how much we're seeing because we have E. coli hanging out in our guts. We have for contact. So you're right. There's a reason we don't have memory like a B and T cell, for sure.
That's right.
And so what I'll talk about a little bit later towards the end is kind of, why this matters for these different models and how it can be used, how this information can be used. So moving on in the paper, what they wanted to also ask, and I'll get your guys' opinion on this because I'm not quite sure. So I really felt that the previous two adaptive transfer models where you're transferring in the first part naive cells only in the previously challenged animals, or you're transferring in cells of both types, the PBS and LPS, into both PBS and LPS animals. I thought that was good. I like that. Now this next adaptive transfer, they say that what they're trying to determine is where these cells come from. So the cells that are responding, where they come from, are they descendants of the cells, or are they new cells that migrated to the spleen after systemic inflammation? I'm not sure if we really answered this question by this model, but maybe I can be convinced if you guys have ideas. And so what they did in this next system is they used traceable congenic NK cells. So traceable, they were able to basically they labeled them with a dye. And so now you can tell the difference between traceable NK cells between the two different congenic strains, the CD45s 0.1 and 0.2. So two different ways of trying to trace these cells. They transferred the traceable congenic NK cells into recipient mice 1 day prior to the LPS challenge. They followed them through day 14. However, they did an additional transfer of naive NK cells 7 days after the LPS challenge in order to have kind of this control naive cells by day 14 in the same recipient mouse. So I hope I explained that well. So they transferred into the original mouse before any type of LPS challenge, traceable cells. So you have transferred cells and you have endogenous cells in the same mouse. That mouse gets challenged with the LPS or PBS, and you had these two different types of mice with traceable cells in it, and then you transfer in a third type, which are from the 0.2 mice, the other congenic strain that are naive. And then you're asking the question, let's look at these three different types: pre-transferred, endogenous PBS or LPS day 14, or post-transferred. And so
what I liked about this was that they could see all three types, right? So they took this, for those who are immunology aficionados. I don't know that I had seen this done before, but this is really cool. The first set were CD45.1, the second set were CD45.2, and they went into a 1-2 host. So they were het. So you could see the original and the transferred using just CD45.1 and 2. So they either expressed one, the other, or both, which I thought that was really cool.
Yeah, no, I agree. I thought it was very cool. And then, you could try to look, you have the ability to look at they're looking here actually at 4 different types of cells and seeing the differences. Right. And so what they saw at day 14 was that there were no differences in any of the mice given PBS originally. So, of course, there's only 3 mice there per time point. So I would have liked to see maybe a couple more mice. But when you look at the animals that were originally given, that were challenged with LPS, but originally given the PBS treatment, no changes, but only in the mice that had previously seen LPS, did you see significantly, I'm going to get little quotes here because it's only 1 star, higher amounts of interferon gamma in the endogenous, first of all, the mouse's own NK cells that saw LPS, and then in the pre-transferred NK cells that also saw LPS early. So the only ones that did not have the increase were the post-transferred naive NK cells. I think this is a nice way to further show us that these cells have a trained response. I don't know if we learned about their origin, because in my mind, I think in the original transfer, when they first transferred in naive cells to het mice, I would think that the transferred and the endogenous, they all started at the same points. I just think you would have had to have cells that have divided or seen LPS transfer in the beginning. I don't know. What do y'all think? I don't know if we're learning anything about whether it comes from descendants of cells or cells that migrated in.
I think the only way to truly show descendants is when you have fate mapping.
Okay, thank you. Yeah, that's what
this is close because it's saying which population of cells is proliferating. And since you have ones that have never seen or have only seen LPS or were seen LPS and then not LPS or moved, there's all the different combinations that I think if you see one population proliferating more or less, you could make some sort of
comment about
conclusion, that. But yeah, I don't think unless you have fate mapping that you could really say that.
Okay.
They seem to be very definitive, though.
Yeah, they do.
They say the results demonstrate that.
Yeah.
This is they have to they get the memory only after the disease, right? Endotoxemia.
Yeah, endotoxemia, which I would agree. I think all the different adaptive transfer models do consistently show that to varying degrees. I would have liked to see some other type of readout because of what Cindy mentioned with the differences in interferon gamma. So they also, one of the questions is, well, how, if the NK cells are memory-like and they are only responding in mice, if they're responding to LPS challenge only in mice that have seen LPS in the past, how is that happening? And so, of course, from the beginning of the paper, my mind's going, well, toll-like receptors, of course, because that's how LPS responds through cells. It's able to TLR2 and 4, always 4, sometimes 2. Well, TLR2 can be both for gram-negative and gram-positive, but TLR4 for sure. So what they did was they, by pre-transferring in TLR2 and 4 knockout NK cells, they found that both the primary, quote unquote, and memory-like response during the LPS challenge were actually independent of TLR2 and 4. So they didn't see the differences they expected to see. And that was big...
So here's the thing, though. These cells respond to the cytokines, right? And so right. The way they designed the experiment, unless I missed something, was that the NK cells themselves are either 2 or 4 deficient. And so that would require the NK cell to be responding directly to the microbial structure, which may not be the case. It may be responding to the cytokine environment.
Yes, right. Which we don't really get to learn much about. It must be a cytokine other than 15 and 18, though, because when they give 15 and 18, they don't see a difference. It's very specific to LPS. In addition. So I agree, though. I think what this suggests is that it has to be a cytokine involved, if it's independent of these TLRs.
Yeah. So the one thing that I wrote down at this point that they did not address at all is, what about the IL-15 receptor expression? And so if it's responding to IL-15 and it's only when IL-15 is there versus when IL-12 and IL-18 from that very first ex vivo experiment, could it just be regulation of the IL-15 receptor, which also has to do with proliferation too? Yeah. So I don't know.
Yeah, definitely.
Not sure.
I agree, because when I'm talking about these stimulations they do, either ex vivo, mostly ex vivo, is these cells are only responding when you're giving the combination of activating NK cell cytokines, IL-15 and IL-18, with the LPS. If you treat those cells alone with either LPS or those two cytokines, they don't have any response. So there's something, I think, with this paper, and maybe if they were able, I know this is challenging, so it's like asking a lot, but if they're able to define that, then of course, maybe, cell nature science is where this goes. But we're still like revolving around that question of, how, like, what are the receptors? How is this working? So we'll actually get into a little bit of how this is working in the next section, but I think the receptor part is to be determined.
Mm-hmm.
They also do some proliferation studies, and they show that the memory-like NK cells that were from the mouse treated with LPS at day 14, they do undergo salvational rounds of proliferation. So they do that by doing CFSE, which when it divides, it dilutes out, and you can see if it's proliferating. So I would say that the summary of all of those experiments is that when a mouse has seen LPS, 14 days later, the NK cells can respond more robustly to LPS, memory-like. So then the next thing they do is they try to get at this question of mechanism. And one of the things about innate immune cells that display this trained memory is they rely on chromatin modifications, and chromatin modifications can be things on histones like methylations or acetylations, which can either help open up the region of the DNA where your gene is going to be transcribed, and it can either enhance that or inhibit that. What's interesting is methylations are a lot of times associated with a decrease in the translation of different genes. But what they were looking at was the interferon gamma promoter. So obviously looking at interferon gamma because that's their readout for all of these studies. And they use something called, let me go back. CHIP, PCR. And basically what they can do is they can fix the chromatin, and they can chop it up, and they spin it down. They can use antibodies to try to detect what modifications were on different histones. And you can use primers of the different, the promoter, the genes that you're interested in. They are looking at interferon gamma. And they looked at a couple different ones. And the only one that they found to increase in animals that were given LPS in NK cells was what I had mentioned before, which was the histone 3 lysine 4 monomethylation.
I think you should say that you can use antibodies that are specific for different methyls and different amino acids. That's the key here, right? You could just buy an antibody, for example, that recognizes this particular mod or whatever else you want to do.
Yes, sorry. Yep.
And you pull down the chromatin with that, and then you sequence what's associated with it, right?
Right. And so this one particular monomethylation, they showed to be increased. And so then what they did was, well, let's remove it. And so they use a chemical that removes. Now, this chemical removes all methylations, correct? It's a global, has effect globally.
It's a methyltransferase inhibitor, yeah.
Yes. So it's going to remove both at that location and other locations. But what they found is that when they treated cells before they adaptively transferred them into the mice, that it abrogated the same type of memory-like response that they had seen in previous experiments. And so what they then suggest from that section is that this methylation in particular is associated with the NK cell memory after endotoxemia. And it's called, the pronunciation I think is synfungin. Mm-hmm. You guys agree?
So the idea is that this mark, this H3K4 methyl 1 is what is participating in memory, right?
That's correct. Yeah. So we don't learn much about maybe the mechanisms of the receptor, but what we know is that these innate cells are using this type of epigenetic modulation for the mechanism of these memory-like cells.
Are you surprised that this inhibitor, well, I guess as you said, it's got a broad effect, so other things could be going on too?
Yeah, I think so.
Yeah.
Yeah, it's not specific to this one methylation on histone 3 lysine 4. But they're drawing the comparison between, hey, we saw it increase only in the LPS. So if we remove it, but that doesn't rule out
other things too. So this was involved most likely, although one could just say it's a bystander, right?
Yeah.
Maybe something else is important, even though, I mean, we're looking at one specific promoter, right?
Well, one promoter, right? So a lot of other different genes we could be looking at related to NK cells.
So do you think that interferon gamma is all that's involved in resistance to endotoxin?
No.
No, right?
No. I think that it's very a lot more complex and nuanced in this. Yeah. But in this model of LPS-induced endotoxemia, they can say, well, you know what though, even with that, because it's a global methyltransferase inhibitor, I don't know. The title is supports, so we can't say that. I was just saying the title might be a little strong.
But they could certainly do CHIP with this inhibitor and see what else is affected, and if any of the genes are relevant to an LPS effect, right?
Sure.
You know, if they're in some other protein, has nothing to do with the immune response, fine. But if they're in other immune-related genes, then that would be cool. So you can do that kind of global stuff, right?
Right. Yeah, with the TAC-seq.
Yeah.
Mm-hmm.
Right. So a little bit then at the end is they introduced a protection model. So they did bacterial infection where they transferred in highly purified cells from either animals that had seen PBS or LPS into naive mice and then infected them with E. coli 2 days after transfer. And they quantified the bacterial burden and looked at the cells that are infiltrating. And so they used at one point in luminescence bacteria that expressed. So you can actually see in the mice, I don't know if that's infrared. Let's see what that type of...
It's light.
It's light luminescence.
Light. Okay. Thank you. You can see there's a lot more bacteria in the mice that were transferred, NK cells that had seen PBS, than the NK cells that had seen LPS. And so what that suggests, both by the survival curve and the amount of infiltrating cells, is that, and interferon gamma secretion, although it's not hugely significantly different, but it was protective against LPS challenge. And
these are 14-day treatments, right? 14 days after treatment with LPS?
14. Yep, correct. Yeah. So not the acute cells, but the 14 days that they had been working with previously, to say now the survival curve is not significantly different, but they did save a couple extra mice.
Yeah.
So is it convincing? There was a reduction. I see reduction, but there's no the luminescence was reduced, but did we do bacterial counts?
Yeah, they did, and it's a little bit.
Yeah, right. Yeah, I see CFU counts, right?
It's a pretty small difference.
Yes, it is.
Yeah. So I think, what this challenge model can tell us is that NK cells probably play a role, and maybe a little bit of interferon gamma goes a long way. But in sepsis or these bacterial models, we need all the cells working together, B and T cells, to really clear the infection.
I think it's a lot to ask just NK cells to take care of this, right?
Right. But it does seem to play a role. What's interesting, though, I think, is that when they tried an unrelated pathogen, so Streptococcus pneumoniae, it did not work, which was evidence from what I was previously saying. If you do a cecal ligation puncture model, you might not see you might see, because you would see E. coli, there would be E. coli in the gut, but anything polymicrobial maybe might not be specific enough. So what it is about LPS, I think that's a lot of work to be done, because an unrelated pathogen did not work.
You should mention the drug experiment in this model. I don't think you did, right?
I don't think I did. Yeah.
It's cool.
In the bacterial model.
Yeah, they used the methylation inhibitor.
Yeah.
And they reversed the phenotype.
Yeah, I'm sorry. I thought you were talking about something different. Yes. If they used, similar to the adaptive transfer studies, if they used that inhibitor, the synfungin, it did not reduce bacterial counts or cause the increase, they say cellular infiltrate, but basically more, natural killer cells, more monocytes coming to the area to fight the infection, which could be because of a decreased amount of responses, so cytokines being produced by those synfungin-treated cells. Yes, thank you.
I wonder if they went out to 9 weeks, and
I don't think.
Then maybe they didn't. It didn't work anymore because it's too weak.
Yeah, I have a feeling it wouldn't have worked just because these are probably the best results we're going to get.
Yeah,
and they're modest-ish. They're fine, but maybe not at 9 weeks.
What is the effect of this methyl mark on the promoter? Do we know on that particular interferon gamma? Is it enhancing?
Enhancing, yeah. So
actually, this is an enhancer mark, right? Not a promoter, correct?
Sorry, yeah, it's an enhancing mark, and it has been shown in previous innate memory-like and secretion interferon gamma to play a role. So I think that's why they focused on it once they saw that it was increased. So, yeah.
I think it's with all the caveats, I think it's still pretty interesting.
Yeah. I mean, I think our job is to, of course, be critical and help. I like what Cindy said. If they're, when trainees are listening, it's good to know what to probe for in different papers. But I think that they still were able to be robust in what they were seeing with these NK cells. Thinking bigger picture in sepsis immunology, I think the focus then is, how do these cells, because sepsis, I guess if it's long-term, how can you try to get these cells activated to try to help clear the bacteria? But then I think it's good. We don't really know what the stimulant is or the receptors involved yet.
Yeah.
Right. To know how to stimulate these NK cells. Because obviously we're always going to be working with them in the splenic environment, which is immunosuppressive by day 14. Yep. So I think there's a lot to learn. This seems like this is not a highly investigated model within NK cell memory, relatively new. I think we mentioned that.
I want to point out that this methyl mark is 22 kilobases upstream of the promoter.
Yes.
Wow.
So it's an enhancer which, wraps around, the DNA wraps around and brings this in proximity to the promoter, but that's what an enhancer is. It can work at a distance.
Right. Yeah, you can picture like a tail swinging around.
Yep. The other thing here, so they have this sentence which we've touched on before, but let me just read it. It says, our results are surprising because the consensus in the literature is that NK cell functions are impaired during and briefly following acute episodes of sepsis. And that was this whole environment thing, right?
Right.
When the NK cells are in the mouse, then maybe they're impaired, but you can take them out and they're no longer impaired. But apparently, despite that finding, if you infect them with bacteria, they can also be partially protective. So,
right. And so how can we use what we've learned from this cell subset towards treating things like sepsis, I think is the goal, because I don't think we can ever rely on the NK cells alone. It's going to be a combination of that. And
they say that NK cells will be targets for immunotherapy. So what would that mean?
Trying to think of that.
Yeah. I mean, I know that they're really, in the cancer world, are focused on NK cells because if you can unlock the suppression and get them to kill tumor cells, because there's also inhibitory receptors on NK cells and the tumor cells can confuse them. And so then they don't eat them.
Yeah.
They can express MHC1, which the NK cells can recognize by their KIR receptors. Mm-hmm. So, I don't know.
I guess if you can mimic the effects of the LPS, training without actually exposing the LPS, you could do something.
Yeah, you can introduce the methyl mark.
Right.
Without needing LPS, yeah.
I just wonder, though, I mean, you're talking about one gene, and I'm sure that particular methyl mark on many other genes are going to have a vast number of effects on the body.
Can you direct the methylase can you direct the methylase to a specific site by hooking it up to an oligonucleotide?
CRISPR, probably.
Oh, yeah.
I don't know if anybody's ever done that.
But that would be one way of making it just go to one.
Sure.
Yeah.
You could target it.
Yeah. That would be right now what we know the only way, since we don't really have surface receptors we can go after.
Mm-hmm. Mm-hmm. Right.
So, yeah.
The problem that I see, though, is that, again, this is a mouse model. And presumably, though, this mouse is eating and you get postprandial LPS spikes in the blood. And so that they're always having some certain level of LPS in the blood. But I wonder how that compares to human, because I've not gone and looked at that or see if that's in the literature, what the difference is between the LPS spikes in human versus mouse. Because certainly it's known that humans also have, after you eat, there's some permeabilization of your intestine and you get some spike in LPS and bacteria that get cleared pretty quickly. Right. I just wonder what role these cells play in that, and this continuous kind of like little tickling, right? So you get a little bit more LPS, you tickle it. You get a little bit more LPS, you tickle it. Pretty much like multiple times a day, this is going to happen.
Yeah, kind of like a tonic.
Right.
Signal.
So where do you draw the line between what is induction of memory versus just over time, just kind of keeping it at a certain level by constantly triggering it? I don't know.
Yeah, right. Like what's the threshold between what these NK cells see as enough LPS to respond with greater interferon gamma?
Yeah.
Yeah. So a lot of questions. I think I enjoyed this paper. I do think it was, well written. Questions to be asked, and hopefully we'll follow up. I did ask Orhan what his career goals and next steps, just to kind of see what he's interested in, if he still plans on pursuing NK cell biology. And he said, I definitely plan to further pursue NK cell biology and innate memory responses. It is fascinating. And currently I'm looking for funding for my next postdoc. So we're, that's a theme, right? Constantly looking for funding. But yeah. Any other thoughts on our paper?
Do you have any more stuff from the lead author that you want to tell us?
Well, sure. So I asked a couple different questions. I had asked about the cecal puncture model and asked if, there would be major differences. And he said, for sure there would be. But they might be more in the acute phase, which is why that model is a bit better for sepsis, because of the acute phase and the cytokines release. And so for long-term effects, he says some flavor of memory would develop after CLP as well, because it is going to involve E. coli and LPS. So it would be interesting to look at that model. I brought up the fact that we did an immune episode on
I was thinking of that. Yeah.
Yeah, we thinking of that. So in 2018, it was published, I think, in the Journal of Immunology. They, found that the NCR1 locus, which is a receptor on NK cells, were different in CD45.1 congenic mice. Yep. And I asked if he thought it had consequences on the results. And even though, the papers definitely prompted rechecking of all the mice, and they said they saw no differences. And he said they felt that they had enough transfer control from PBS mice to naive and vice versa to stand by their results. I don't know what saw no differences means, but I think that that's something to consider when specifically looking at NK cells in this mouse, this model. I asked about the purity of the cells. It wasn't always well stated because they really brought up in the beginning how those first ex vivo and vitro studies were enriched. So whenever you hear enriched, it's like 80 to 85%. There could be contaminants.
Right.
What about that? And so for the co-cultures and activation transfers, he said they regate only on the specific congenic NK cells compared to endogenous controls. And so the contaminants shouldn't factor in. And the transfers for protection, so then the bacterial studies, they said they were done with the highest purity. So that suggests that some of these adaptive transfer models were done with the 80%. But I guess if you gate well enough, I don't know, Cindy, if you have thoughts on that.
Well, I was going to ask you, did you go and look, because I didn't, at the supplementary data at their gating strategy, and were you convinced? Because I haven't worked with NK cells, so I don't know what's the right way to do it and whether their gating strategy was sufficient to claim their purity.
So yes, I did. We are working with NK cells within some of our collaborations. So I kind of tried to compare them to that. So one of the things that's also interesting is that there are things called innate lymphoid cells. These are recently, I'm quoting, but recently discovered innate cells on the lymphoid lineage, not myeloid, but lymphoid lineage that are really defined by cytokines alone and some transcription factors, not really by surface receptors. And so they're plastic. And actually, there's three types. The ILC1s are ones that can, NK cells can be a part of ILC1s. And so you really have to do a good job of making sure that those are not contaminants, but they are a very rare cell type. So I was kind of focused on the ILC1s. I had followed up with him with a couple extra questions. Did you gate on ILC1s to see if you saw enhances in interferon gamma? Because they're very similar. And he says, another cool question, the problem with ILC1s in their transfer or co-culture model was the number of cells. And this is very common with the cell subset. It's hard to isolate them. They're very rare, and they're usually in different tissues. So with big acquisitions, I had enough conventional NK cells to confidently evaluate their responses, but really not enough of these innate lymphoid cells, the type 1, to look at. Thus, it remains an open question. And I agree, adding transcriptional factors would have been better, but it remains to be done. So I had brought up the fact that, looking at transcriptional factors of different cell types would have helped to really define whether they're NK cells or ILCs. So it's just something I think to keep in mind when looking at these cell types and when you're gating on low numbers of cells. I asked also about, right now there's like these three major types of memory like NK cells, and where does their NK cells fit in? And he said, really, it's hard to say because they couldn't pinpoint a specific subpopulation. That's one thing I didn't bring up, but they tried to, by surface receptors, say, was this because of one specific cell type of NK cells? But it didn't matter in the end. So I think for now they don't know. And further investigation is needed. And I think that's it for the questions.
Cool. Thank you, Steph.
Sure.
Yeah, that was fun.
Let's read a couple of emails. Anthony writes, the ending of Predict is deja vu all over again. So Predict was my pick. Well, not Predict, but my pick a couple of episodes ago was the ending of Predict by the U.S. government. Predict was a program to identify new zoonotic diseases. It was established by USAID, and they stopped it. So it's unfortunate.
Right.
Very much so.
Ryan gives us a whole bunch of links about anti-vax movement.
Yeah.
In The Guardian, The Washington Post, he says, apparently there was a study, according to The Guardian, examining political ads on Facebook. In this case, the findings showed that RFK Jr. and Larry Cook got called out for producing the majority of anti-vax ads on the Facebook venue. In past episodes on Twiv and Immune, they pointed to Dale Bigtree, Bernard Seltz, and Andrew Wakefield for spreading the majority of anti-vax political ads via movie promotion on Vax the Movie and various political campaigns over vaccine laws. And then he gives an update on the California vaccine laws up for debate in 2020, SB 276 and 277. I just don't understand why these individuals have such an anti-vaccine agenda. Oh, I know why. They get paid. Sorry.
Yeah, they do get paid. That's right. And we don't get paid to promote.
No,
vaccine.
we don't.
No, we don't get paid. I don't want to be paid because it's the right thing to do, right?
That's right.
So you must have heard about the Italian physician who's now in the news about being a pro-vaccine person in Italy, where vaccination is really in trouble because they have no coordination there. But what did he say? Gasoline is flammable, vaccines are safe and effective, everything else is lies.
I know.
It went viral.
I know. See, that's the thing that can go viral because it's so short and to the point and punchy. But then people get, they want explanation, and that's a lot harder to make viral. We can talk about the science all day long, but it's not as viral.
No, I know. We don't go viral. That's the problem. We don't go viral.
Yeah. So I'm currently reading Bad Advice by Paul Offit. I don't know if you've read it, but he talks a lot. It's really interesting. He talks a lot about the dichotomy between scientists needing to be precise and at the same time allowing the level of uncertainty that we have. Like, for example, you can't prove something without a doubt, right? Mm-hmm. You can support it or you can disprove something, but it's impossible. And so to convey that to someone who isn't in science sometimes is really challenging. And so whenever you say, they say, well, can you come out and say definitively this will never happen?
Right.
A scientist is always going to go, well, no, we can't exactly say that. Here's what we can say. And bam, you're done for, right? Sure.
Right.
And so our training trains us to be exactly our worst advocates for these kinds of things. And so it's interesting because we also attack our own, right? So scientists do get out and speak and do make those sound bites and do leave out a lot of stuff for clarity so that we can support our cause, get trashed by other scientists. They're like, you can't say that, you can't do this. Yeah. And so there's this disconnect and there's this tension. And so how do we solve that? Because we're basically killing science, right?
Yeah, it's a big problem.
I don't know.
Yeah.
I agree. Cindy, can you take that next one?
Ornob writes, Dear Immune Investigators. Did I say that right? This one's probably not taken as it's quite bad. Anyway, regardless of what you call us, I'm a first-year graduate student at NYU. Congratulations. And this is my first time writing to Immune. Congratulations on an amazing podcast, and I can't believe it's already been more than 2 years since you guys started. We can't either. I won't discuss the weather, as Vincent already knows all about it. Because she's in New York City. I'm writing to bring to your attention this new Cell paper on a TLR, my favorite topic. And this paper is by Veit Hornung, and it's on TLR 8. And it says, although you may already have seen it, I'm always amazed by the variety of ways that the innate immune system detects the presence of pathogens. I thought this would be a nice paper with which to go into nucleic acid sensors and PRRs in general, which have not been explored as much on immune. best or not. This is kind of funny. So I don't know if you do this, Vincent, but I tend to try and not pick exactly the papers in my area.
Yeah, sure.
Because
right.
I'm like, otherwise I risk the, going into way too much detail or to just always, pick my favorite things. So I try to diversify out. But, my training is in nucleic acid sensing PRRs, and that's what I the majority of my research on. So I'm very excited about this paper, and I would love to cover something like this. Thank you.
That could be your next paper.
You could.
If you wanted to.
TLR8 is a sensor of RNase T2 degradation products.
Yeah.
So T2, I guess, is the RNase that chops up the RNA so that it can be sensed by TLR8, right? That's the bottom line.
Right. The cool thing about nucleic acid sensing TLRs is they seem to work in endosomes, right?
Yep.
And part of that is, there's acidification. Part of that is proposed to be proteolytic alterations to the protein structure. And then this would also be, for example, you need the nucleic acid, but then you also need it chopped up a certain way in order to be able to bind and activate the receptor. And that would happen in the endosome.
Got it.
Yeah.
Thanks, Ornob. And let's take one more. Steph, can you take the next one?
Sure. It's long. Are you guys ready?
Yeah, it is. Mm-hmm. Yeah.
Okay. All right. Niraj writes, Dear Twee Informants, it's been a while since I wrote, but listening to the latest podcast on the role of fiber in our diet and how the microbiota derives short-chain fatty acid shaped CD8 positive T cell repertoire was totally fascinating and intriguing. Now I know why most late-night joints in NYC close to Rockefeller University, that's where I went to grad school, used to serve extra sticks of celery with delicious chicken wings and beers. And okay, he put parentheses, the most bland food is always the healthiest, ironically. Yes, that's true. But overall, this research is yet again a testimony to how basic research can uncover such intriguing connections, which leave you thinking that diet is playing a very important role in shaping our overall physiology and not just a means to provide energy for our daily routines. Yet again, Mom was right. So I will be eating my veggies more. I personally feel that the modern day lifestyle does compromise the importance of cooking and eating healthy, especially when there is so much packaged food around, which can be ready for consumption with a few clicks on the microwave. But oh well, that's the price we pay for technology and having a life that just seems to be gaining pace with every fleeting moment. But getting back to the research article, even I am very much interested in knowing as to how exactly the short-chain fatty acids are shaping the memory potential of CD8 T cells. Interestingly, though the authors don't delve into it, I feel there are bound to be other types of cells, even non-immune, that are benefiting from this regard in the gut microbiome. I think that's true. Talking of which, it reminds me of a person I met in the park the other day while my son was playing, and he mentioned, who needs bacteria and all that filth for anything? To which I responded, almost everyone, as otherwise you might cease to exist. He gave me a confused look and asked why I was suggesting that filth and dirt are good. I thought I should tell him because you were covered in it. But in the interest of sanity and not freaking someone out, I told him about some of the health benefits and the reason why there are so many probiotics on the market. We owe a hell of a lot to our microbiota. And overall, I feel too often people associate microbes only with the disease origin, propagation, which is partly justified. But looking at the plethora of scientific research in the last decade or more, I feel we are still in the infancy of this field, and next several decades will help us unearth more fascinating aspects of how our bug buddies shaping our physiology and probably behavior as well. In the end, I just wanted to congratulate you folks for taking out the time to spin these jewels out. Wow, that's so nice, jewels. So keep up the great work, and especially for Professor Racaniello, here's to many more years and decades of podcasting and spreading the wisdom. Ironically, I'm struggling to keep up with your podcasting pace, and my after-dinner walks are getting longer and longer, as there is just more and more intriguing and relevant research to indulge in and learn from. I personally can't thank you enough for your efforts, and it's only appropriate that you were bestowed with the Award for Education by the American Society of Microbiology this year. It's a noble cause that you have initiated, and it will benefit many young scientists and curious minds who sometimes feel lost in the wilderness while trying to pursue their passion for research. Personally, the excitement of deciphering the unknown is the biggest thrill of all for me. So I hope the width of the Twix podcast series will continue to flourish and expand the dimensions of my knowledge. Best, Niraj. That was
so sweet.
That was awesome.
Very nice.
I had two short comments to that. One is the comment that there are bound to be other cell types that are benefiting in this regard from the gut microbiome. One of the papers that I was reading with great interest recently was how butyrate produced by the microbiota regulates macrophages' ability to kill bacteria. So absolutely, no doubt. And the other thing is we already know is a definite connection between microbiota and behavior. Right. Again, through production of a variety of different metabolites. So that gut-brain axis is a big thing recently. And I'll say, I'm going to ask you guys, do you cook a lot of your own food?
Yeah, I do.
In college, I did not. I try to more now, but it is tough, especially if you're working late. It takes a lot of prep. I know that if I can prep on Sundays, I do better.
Yeah, I do. I've gotten really good at trying to research some options to simplify making meals and use fresh ingredients and things that take, about 30 minutes to prep. Because when I'm here and I get home, I have 2 kids at home, and they're like teenagers and preteen now, so they're starving by the time I get home. And I don't keep junk food in the house. They're like, when's dinner? I'm like, I just walked in the door. I need 30 minutes. And it's like, set the table, just start your homework, and it'll be done. Right. But yeah, I try as best I can to really cook with fresh ingredients and cook regular meals, because it's true. When we were, over the holidays, we went and visited our family. And so there was a lot of ordering Chinese and eating pizza and eating out. Oh my goodness. Just after 3 days of eating like that, I felt horrible.
Yeah.
And, detoxing myself from that and eating healthy food again, it really, really changes how you exist. And I don't think enough people think about that, how much added sugar and crap is in the food that you get if you eat out or if you buy packaged goods. Yeah. Anyway, I will stop on my soapbox.
I was home for 2 weeks. My wife had surgery in December, so I stayed home and I cooked. And I actually got really into it. I never did before because of the prep. I don't really like the prep, but I ordered a bunch of Blue Apron meals.
Oh, yeah.
So you have everything right there, and it's pretty healthy. And we got all vegetarian, non-meat entrees, right? Fish and vegetable. And it was really good. I enjoyed it. I enjoyed getting there and putting out the stuff and cutting it and chopping it and frying and it was fun. So I'm
kind of meditative almost, it's that transition coming from, brain going all day, at the lab and everything. And you come home and you kind of calmly do this thing that doesn't require a lot of brain power besides don't cut your fingers off. And then,
it's a zen. You definitely get into it and you just focus. And then in the end, you have something you made and it's actually good. I think that was pretty cool. It's a lot of fun.
Yeah.
So I enjoyed doing that. And so, I'm more, and now our kids are older, so we don't have to cook for them, and it's more relaxed. You don't.
Oh yes, very much more so. When you when's dinner?
Can be more focused on healthier stuff for sure. Definitely.
Yeah.
All right, let's do some picks. Steph, what do you have for us?
Okay, yeah. So there was a paper in PLOS Medicine that came out where they had looked at a bunch of clinical studies, and they had shown that when oral poliovirus was given at the same time as rotavirix, which is a monovalent vaccine for rotavirus, that combination was a contributor to reduced IgA seroconversion to the rotavirus vaccine. And we have so this has been suggested in the past. There's been evidence of this in previous studies, but I kind of like it was like the combining of two worlds. So Vince, I know you worked on poliovirus. We're working on rotavirus-induced immunity here in Sally Permar's lab at Duke. And so it was just nice to see such a big study that demonstrated this and in clinical trials that they were able to reanalyze. And so what this means, though, is that potentially where this vaccine is needed the most, where rotavirus deaths are the highest is countries in Africa and Southeast Asia that also get polio, that when they start, so they're going to start, I don't know if discontinuing is the word, removing the oral polio vaccine from the vaccine schedule. Mm-hmm. And Vincent, from my understanding from listening to your podcast, is that's because then you're secreting it's live. You're secreting polio and then you have polio in the environment. And so by removing that, maybe we will then see a boost in rotavirus seroconversion, IgA, which is important, in these very important areas of the world that need this vaccine to work.
So they're going to remove it at some point, but it has to be replaced with an inactivated vaccine because we're not ready to stop immunizing entirely. But
right.
Yes, sorry.
This is really interesting because for years we worried that other enteric infections were blocking OPV uptake. Right? And now it turns out that OPV could block rota. It would be really interesting to study that in the mouse model, you know. Is there a mouse model for rotavirus?
Oh, there's not a infection model. So pigs, so what I had worked with in my PhD, the gnotobiotic piglet model is the best model for infection because you get full clinical signs, you get robust replication. But polio in pigs, have we done that? It'd be interesting.
No, it won't go because they don't have
because
receptor. Yeah, you'd have to make transgenic pigs.
I could make transgenic pigs, right?
And probably not worth it.
But in this, probably not. But in this paper, they talk a lot about just the results, but they don't really delve into the mechanisms. And so one of the things I think about is why would that be happening? And it must be that the polio vaccine maybe is more immunogenic and is taking up more of that capacity of those B cells in the germinal center of the gut to pump out IgA. Maybe.
It could be, or it could be at a virus level. It could be interference. Yeah. So when you get OPV, OPV is trivalent. The first time you largely seroconvert against one of the serotypes because the other two it interferes with the other two. And in fact, the doses are adjusted to try and get around that. But that's why you need 3 doses. So eventually you will get seroconversion against all 3 serotypes. So even amongst itself, the OPV interferes.
Do we still have all 3? Because I thought one of them was eradicated.
So the WHO is using bivalent now, type 1 and 3, because 2 has been eradicated. Correct. Okay. Actually, 2 and 3 have been eradicated, but 2 was declared eradicated 5 years ago, and so they stopped putting OPV2 in the vaccine. But the problem is, a consequence of that is now we have vaccine-derived type 2 outbreak more than polio. We had more cases of type 2 polio from the vaccine last year than wild polio. And so
I saw that.
The WHO is saying you have to use IPV, which is trivalent, but there has been a shortage of IPV, so we couldn't use that.
And is that cold chain? Does it require?
Yes, it does. It has to be frozen. They both. There is a new OPV that's been developed, and it's in clinical trials that looks promising because it doesn't revert, and they may substitute that in for the OPV2. We'll see if they're going to do that or not. But it's a very interesting situation. This is a cool paper. I wasn't aware of that. Yeah. Cool. Cindy, what do you have for us?
So I have a website that is called Diversify Immunology, and this is a shout out to Kathy Spindler. I was on an email chain where she reminded you that had to be your next pick on immune, but when you didn't have that, then I put it up.
I forgot.
I picked it on another podcast, which was right after that email. Yeah.
Okay.
I forgot that. Yeah, good.
So this thing is called the Diversify Immunology Initiative. And on the main site, it just basically says that, studies have shown that more diverse groups work better than, non-diverse groups. And that other studies have shown that a more diverse group is generated when candidates are identified from a list rather than based on recall. And I would agree with this because I know, for example, when we put our seminar series speakers together, you sit down and they send you an email and they said, by Friday, let us know the names of 5 people or whatever. And so your brain goes to like the last 5 papers you read or whatever. Right. And so you don't really have a good breadth of individuals, especially diverse individuals. And for me, it's a real passion because I have a program that's really based on diversifying the faculty and, making sure that you have those role models, people who represent all the different diverse races and gender distributions, et cetera, coming and giving seminars on science. Our students are diverse now. They're more diverse. And so if they don't see that diversity reflected in our seminar speakers, they have a hard time envisioning themselves as, faculty in the future. And so this list, what it does is you can self-nominate. There are only self-nominations. And so you go and you sign up. And so I did. Have you done that stuff yet?
No, I haven't yet,
okay,
but I'm going to, I always get a little bit like, what's the term? Because I'm like, I'm just a postdoc. I don't need to be on there, but I should put myself on there.
You should put yourself on there.
So it's really my lab. We're pretty diverse postdoc crew.
Right. So it's twofold. You want to put your name on that list if you are, a woman or LGBTQIA or disability or minority. But also what it is for symposium organizers, award committees, search committees, they can go and say, we need to make sure that we have a diverse, group of people. Where can I find a list of people? And so AAI had one list like that at some point. I'm not sure if it's still there. I know I put my name on it a while ago, but this now list hopefully will expand. And so you just go. It's a very easy process. It takes like 2 minutes to fill out the form. And then your name will be on the list. And hopefully you can spread the word, and people who are looking for seminar speakers or, people to nominate, they can at least go there and say, oh, okay, are any of these names ring a bell? Because it may be like, Dr. Leifer, I wouldn't have thought of her, but yeah, that's right. I can ask her, whatever, so I think it's really great. And so the list is maintained by Rebecca Pollitt. And so I encourage you to go check it out if you're in the area of immunology and you'd like to get some exposure. And also for those people who are maybe listening who are on search committees and so forth, go and look there for some resources to help keep your candidate pools really diverse.
Yes, nice. I'll share it.
Thanks.
Cool. All right. What did I do it on? What podcast?
Probably TWIV. No?
Yeah, maybe. I know we didn't do any TWIVs over the holidays.
She sent it on November 26th.
Maybe. I picked it on something. Yeah. Anyway, my pick isn't... Actually, we were supposed to do this podcast last week, and this was more timely last week. But
good talk.
It's a New York Times article called "There's No Winter Break from Publish or Perish." Mm-hmm. An analysis of submissions to 2 top journals showed that scientists in the U.S. were highly likely to be working during the holidays. And I think that's really funny. The rest of the article is exploring different cultures, who's working during the holidays. But submissions is really meaningless because the work was all done weeks and months ago.
Oh, yeah. Right.
So submitting a paper, you could do it at home, right? Just click a button. Anyway, it's a fun article. And the picture is great. It's a picture of Einstein writing something with 2 Christmas trees background.
Oh, that's really too funny. I see that. Yeah.
And this reminds me of, they say here, some countries, people never work over the holidays. Others, they do. And I had emailed some people in Denmark in, I think, August. And I got this reply. Oh, we're all on vacation now. We can't answer you till September. It's funny. It's really fun. Email makes it easy to work anywhere now, right? But some people do not. Dangerous. Some people do not want to, and I understand that they don't want to do that. But many of us continue to respond and so forth. So anyway, it's a fun article. All right, that is Immune number 27. You can find it on any podcast player on your phone or tablet. We'd like you to subscribe so you get every episode, and we know how many people are listening. But we do have a website where we have links to the articles we talk about in our picks and so forth. That's at microbe.tv/immune. If you really like what we do, we could use your financial help. We do have expenses. Go to microbe.tv/contribute. You can use PayPal or Patreon to give a fixed amount of money once, or you could give us $1 a month or $2 a month. We would love to have that from you. We appreciate your support. Immune is composed of the 3 of us. Cindy Leifer from Cornell University on Twitter. She is Cindy Leifer. Thanks, Cindy.
Thank you.
Steph Langel's at Duke University. Stephanie Langel on the Twitter. Thanks, Steph.
Yeah, this is fun.
And I'm Vincent Racaniello. You can find me at virology.ws. And who's next on this? Am I next, or is Cindy? Cindy went last time. She did the measles. Immune erasing. So I'm next.
Woohoo.
All right. I'll have to come up with something that is not related to what I do. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.